So now we are at the final data talk that we're going to talk about. And we're going to talk about how kinetics come to play in all these different situations. And it's really interesting because we have been moving pretty linearly through the history of life. And only late does kinetics really have a major impact on life. Early on, the geological processes don't depend that much on kinetics. But these processes, when we get to advanced multicellular organisms, and by the way, when I say complex, I mean more complex than bacteria, okay? I don't mean J.S. Bach or something like that. I mean like slime molds and on up, okay? But remember that we had oxygen expansions, and this is in the aftermath of that. The Cambrian explosion came right after the oxygen expansions. And I found that paper that I mentioned in the last data talk, by the way, where somebody correlated fossil life expansions, microbursts, with microbursts of oxygen from the geologic record. That's a really exciting paper to me, and it's the kind of thing that I think happened because I think there's a strong link between chemistry and biology. But oxygen even allows new chemistry. In a sense, oxygen allows the cross-links that cause multicellularity. In many cases, the cells are cross-linked with oxygen-containing structures. More oxygen means it's easier to do that chemistry. What's interesting as well is if you look at what cuts the connections between cells when cells move, or when cells, what forms the connections, zinc is usually used to cut, and it's used outside the cell rather than inside. Remember, the inside of every cell still looks ancient. Copper looks like it actually helps make those connections, and it does all sorts of things with oxygen. Pretty much if you're doing something interesting with oxygen and you're human, you're probably doing it with copper somehow. If you think back to biochem, those of you who've taken biochem 2. Biochem 2, what's the last enzyme in the whole oxygen-using series? The enzyme that actually converts oxygen to H2O in the mitochondria. It's a copper-containing enzyme. And so it looks like when you're touching oxygen, you're using copper to react with it. So the thing is, with these new connections being formed and made, with cross-links being made, you have the potential to make organs. You have the potential to link certain kinds of cells together and to say, hey, all of you cells, you all have the same job. And eventually that becomes a liver or a heart or a brain. So different kind of compartments of cells put together can hold different chemicals. And there's also subcellular compartments, different compartments inside the cell. And it looks like oxygen allowed both of these kind of compartments to form. As we look through this, you can say this is definitely the physicist's way of looking at things. You can say that we have these scales and we have these cells are a specific kind of compartment. And we have compartments bigger than cells that we call organs, compartments smaller than cells that we call subcellular structures, or vesicles, or whatever it is, mitochondria, plastids, they all fall into the same category. And you could argue that the size of these things, the physical size of these things, is related to the chemistry that they have to do. One of the examples for new uses of copper and zinc, you will see the same job being done by iron in bacteria and by copper and zinc in humans. For example, superoxide dismutase. It goes from an iron manganese protein in prokaryotes to a copper-zinc protein in animals. Making the extracellular cross-links, of course, you don't have any in the smaller stuff, the unicellular stuff. And then you start off making them with manganese and iron in plants, but then in animals, you end up using iron and copper to make those. And those are usually oxygen-involving chemistry. Even the inorganic messengers, you'll see calcium and zinc are used for brain chemistry. They're used for plant signaling. They're used for multicellular talk, whereas for single cellular stuff, you see phosphate, iron, and manganese. Okay, so we have lots of interesting things. And the other thing is communication between cells is done by organics oxidized by iron and copper, like adrenaline. Adrenaline is made by iron and copper working with oxygen on an older molecule that was used for a different purpose. When you think of the molecules, small molecules like adrenaline, that send a signal like that? What would be the general form of that communicate between organs and coordinate processes like multicellular development? What class of small compounds that we can give as drugs and that we can make as organic chemists that would fall under that communication category? Does anything come to mind? Yeah. Estrogen, progesterone, testosterone. All these things are actually really accessible. Now, there are peptide and protein hormones as well. It looks like you can do multiple ways to communicate, just like we can have different communication forms now. You can call on the phone or you can text. But you do have this pattern that it seems like copper and zinc keep showing up for advanced purposes. So you do have some exceptions that you can think about. You can raise your hand and say, well, Dr. McFarland, what about this example? Anybody work with Dr. Ridgway ever? His snails? He has a whole bunch of snails, or had. He's retiring now. And the snails have blue blood. They're copper-based rather than iron-based. It works for them. And in fact, if you ever watch Star Trek, you know who this guy is. Not Zachary Quinto. This is Leonard Nimoy. Mr. Spock. Not Dr. Spock, by the way. You got to be accurate in this class. Okay. Anybody know what kind of blood Mr. Spock and his species have? Right. Probably because, I don't know, maybe it got past the censors better on the 60s. Basically, or maybe they ran out of red paint. That's true, so. But like when he gets a cut, it will be green blood. I think you see that in like the new Star Trek movies as well, because it's a huge thing. Vulcans have green blood. The reason why they did this is they knew that there were things like snails and octopi that use copper-based blood, and so they said, let's have a humanoid that uses copper-based blood. Now, hemocyanin, which is the copper-based oxygen-binding molecule, actually will bind very tightly, has a big K. It works well with low oxygen in cold environments, like if you're at the bottom of the ocean. Okay? Oxygen goes on quickly and it comes off very slowly at those temperatures, though. It's good for binding, but it's not necessarily good for transport because it never lets go. So, as a scientist and as someone who loves Star Trek, what is my problem? And you know I have nothing against the color, okay? And yet I still object to Mr. Spock having green blood. Why? Well, the one thing is, here's a picture of Vulcan. It's a little washed out. It's a desert planet. Okay, it looks like Dune or Arrakis, isn't that? No. I forget the name of the planet in Dune. But anyways, it looks like Tatooine. Let's say Tatooine. Okay, we know that's a desert planet, or Jakku. Why am I bothered by this fact? On a desert planet, those are kind of different thermodynamics than at the bottom of the ocean. It looks like copper might work well for some organisms, but it would be better if it came from the Boba Fett planet, the Genosis or whatever it was, a cold water-based planet, and if he was an aquatic series. Of course, they weren't thinking about this. They were just putting green makeup on him when they first came up with this. But it does bother me, and I say that as a Trekker. Real Trekkers call themselves Trekkers and not Trekkies. So, anyways, you can talk to my high school friends about that. Okay. So all that is to say, iron and copper, it looks like copper is used for certain functions, but like we don't use copper for blood, A, there's not enough of it, B, its kinetics are not right for a atmospheric room temperature situation. So if you think about endosymbiosis, and actually we are making steps in this, but there's still a lot we don't know about exactly how this happened and how many times this happened. We talked a little bit about this the other day. But I want you to realize this is formation of a subcellular compartment that has a certain dimension. Mitochondria are really small. Eukaryotic cells are much bigger than bacterial cells. And that's one of the things about why we think this happened, because they're the right size to be like bacterial size. The main thing is they're small compartments, and they have distinct chemistry that shows up in their distinct colors. Iron is red. Magnesium in chlorophyll is green. The color goes with the function. And that goes with the kind of element that you expect to see predominant in it. So you all know that mitochondria make ATP, and they breathe in oxygen. It's where all that does. Chloroplasts actually do the reverse chemistry. They're not related at all, but they're used in a sort of opposite kind of way. Chloroplasts are used to make oxygen. Mitochondria are used to burn oxygen. Both of these have their own DNA, their own membrane, their own lipids. They even bud when the cell divides, like they're bacteria growing in the cytoplasm. And because we know that some bacteria, different unicellular organisms, can phagocytose each other, maybe one stuck around. Again, this is sort of a follow-up on the question that we talked about this earlier. So the one thing to remember is that both the chloroplast and the mitochondria are oxygen-rich zones. So not only are they unusually sized compartments and they have their own elements, they also have their own oxygen profile. And that's another argument for why, oh, maybe they developed later, because it looks like the cytoplasm is sort of frozen in time. The cytoplasm is reducing. And even though we depend on oxygen, it's important to remember the part of your cell that depends on oxygen is where the oxygen goes. The rest of the oxygen is actually, the rest of the cell is actually low in oxygen. In fact, it looks like you're deliberately keeping the oxygen out of the cytoplasm, because when it's cut loose there, it starts to do damage. Just like we're theorizing, oxygen, when it first appeared, would start to do damage. This is the way that, for example, and this extends to other compartments, like the peroxisomes. Did you talk about peroxisomes at all when you talked about lipid metabolism in Biochem 2? Anyone vaguely remember peroxisomes? It's like the Lorenzo's Oil movie is actually about a boy with a peroxisome defect. And peroxisomes are kind of cool because you can see it in their name. They use oxygen to do chemistry. They burn the long fats, and they also will make certain molecules. But they're like mitochondria. Special oxygen chemistry requires its own room. Just like we have to have biohazard levels. It looks like oxygen is a hazard to the cell, and therefore you use these compartments to use this new oxygen that came onto the scene. So these different compartments, once you establish them, you can link cells with oxygen. You can put oxygen chemistry in its own little room outside the cytoplasm. Once you have the room set up, you've got to be able to talk between them. You've got to have doors that go between the room. Now, we don't have literal doors, but instead we have chemicals, because everything is chemistry at this level. Isn't that cool? So if you look at the ways that things communicate between each other, you actually also see a progression. If you put more and more complex organisms in order, you'll see that more and more messengers, it looks like over time you get more and more messengers. Right here, you have calcium, zinc, and copper showing up when you start to go to eukaryotes, when you start to get multiple cells. And if you're wondering about what kind of signals they send, that's all the way on the right. But what I want to point out is when we get to copper and zinc, we're kind of at the part 4 of the story, act 4 of a 5-act play. What's act 5? Sodium, potassium, and chloride. And these are the ones that really depend on kinetics. What I want to say is that these actors that are come in, first you have calcium, which is sort of different from the others and acts different from the others. It's used by single-cell eukaryotes. And multi-cell eukaryotes really start to use the singly charged ions. Not to build structures, but to have particular kinetic responses. Also right there's the organic chemicals like the hormones. Those would start to be using. And you don't use hormones to communicate with the mitochondria. You don't communicate with subcellular things that way. It doesn't actually make sense if you think about it. It makes sense to send them through the blood to another organ. And that's what the hormones do. They coordinate the organs over time through the blood and lymph and all that fun stuff. So the chemist's view of biology can be taken as you have these organs, and some of these organs, like the ones that make the hormones, the ones that make the insulin that coordinates everything, those are also enriched in copper and zinc. So even the whole organ, when you eat liver, liver's not on here, but I know that liver is enriched in iron and copper and zinc and these metals. And if are there people who eat, I think you can eat the glands, right? You call them like sweetbreads or something like that? Anyways, I like liver, and so it seems like I've had those before. I just don't know exactly which organ it was. But I know when I was eating those organs, I was probably eating a lot of copper and zinc, because those advanced functions use the advanced metals. And I'm actually comfortable going that far to say those metals are advanced. Oh, and by the way, if you know about iodine in the thyroid, you know, iodine and the thyroid are really closely interlinked. If you think about what the thyroid does, bacteria don't need a thyroid function. It's involved in a sort of hormone communication, interorganism, multicellular type function. So iodine actually fits into this as well. There just wasn't room to put that in the book. Believe it or not, I did actually cut something. So here are what hormones look like. Here's adrenaline. Here's tryptophan, which looks like serotonin. And here's a plant hormone. Those all look like amino acids that have just been modified a little bit. Few bonds broken, few oxygens added. It looks like if you want to make a messenger that sends very diverse kinds of messages, what you do is you do a little chemistry on an amino acid, make a new shape, send it through the blood, and you use iron, copper, or zinc to do that. In fact, you can even take the older signals and associate them with iron, and the newer signals and associate them with copper. Again, iron's kind of weird, so you can't do it completely, but I believe you can see that trend. And here's the only place where you'll see selenium, but selenium is kind of cool because it has cool sulfur chemistry and it does some cool things with cysteine to make a particular hormone. For the most part, again, you have this trend, and this trend accounts for all sorts of kind of messages, all sorts of kinds of organisms and organs. So you can even take the organ and you can say, what does the organ need to do? Well, because of what it needs to do, it's going to use certain elements to do it. And those are the best elements to use. And so on this planet, and on all planets that will have most of the top of the periodic table, I would expect these things to be followed. You know the iron, that the liver is rich in iron. It's rich in iron for a reason. It does the metabolism that iron is good at. It also keeps the reactive iron in some forms away from other parts of the body. The main thing is when you see fast observations and fast reactions, you get to a part of the periodic table that we haven't talked about yet. You get to the last couple chapters of the book. And that's what we want to talk about. We want to talk about how all that comes together. So this is organized by function, and this is organized by element. And you can actually take the different elements and you can say, oh, which organs do I find it in? And they will tend to go together. You can make a chemical sense over which elements are needed to be used in the liver, spleen, and blood. So again, you can make dietary connections here. And these are not necessarily predominant, but they're like where it would be found most relative to other organs. You know, it's that kind of thing. Okay. And that leads us to a really good stopping point, because once we talk about that, next up is we're going to actually bring in kinetic data, and that's why we had to wait until now to actually talk about that. But I will leave you with that cliffhanger.